All right, we ready? Everybody has their notes set up, right? Okay, good. Okay, I know we slept since, so I'll quickly go over what we already kind of know. We know that when cells divide, they're actually multiplying, right? So when cells split in two, then you end up with two new cells. Now, what's really kind of weird is that those two new cells are actually called daughter cells. I don't know why we don't call them son cells, but they're called daughter cells. So whenever I refer to daughter cells, just know that I'm referring to the two new cells after cell division has taken place. We talked about why cells divide. If you're a one-cell organism, this is the only way you're going to be able to reproduce. If you are still growing, you grow by adding more cells. Some of us get to a stage where we're done growing up, and then we just start growing out, and so that's still cell division as well. Also for repair. So we've all had skin needs and so on and so forth, and we know that the skin is going to divide and replace itself. We talked about all the parts of the cell that are used for cellular division: the nucleus, the cytoskeleton, as well as the centrioles. Remember that the centrioles are only going to be found in animal cells. So there are no centrioles in plant cells. Plant cells don't have them. All right. So we started with talking about the cell cycle, and we drew this. Does everybody have this on the right side? Okay. And we said that the longest part of the cell cycle is called interphase, right? And so interphase has three parts: G1, S, and G2. And the cell spends 90% of its time in interphase. So, and at the end, I'll show you kind of a cross-section of onion root tip cells, and you'll notice that the majority of them are in interphase, because that's usually where the cell is. Okay, so we wrote some information about the interphase, and we drew this picture. So regardless of whether it's G1, S, or G2, interphase looks all the same. If you're looking at it under a microscope, the different phases, G1, S, and G2, all look the same, even though different things are happening in each one. So when you take your test next Friday, what's really important is that you not only know what's happening during these phases, you also need to be able to recognize a picture of each phase, or a diagram of each phase. In interphase, you'll notice that the nuclear membrane, or the nuclear envelope, is well defined. Okay, so you can see it. Okay, so it is all there. You'll also notice that there's still a nucleolus. It still exists. And you'll also start to see the centrioles. Okay, they're called centrosome when they're in pairs, but you'll see the centrioles only in animal cells. And that's what it looks like in interphase. You'll also notice that the chromosomes are kind of stringy looking. Okay, so you can see them, but they're very kind of look very dark. And that's interphase. We talked about the different phases of interphase, the first one being G1. And that's really called the first gap phase. But the way to remember it is just to call it growth, because the cell is just growing and doing its everyday job. Now, at some point, the cell says, you know what? I'm getting way too fat, and I'm going to need to divide because now I'm too big. Okay, or it says, you know what, I don't need to divide. So hopefully you wrote this down. If you didn't, please do so. the cell could decide to go into what's called G0, which means it non-dividing. So no dividing. Now that could be temporary or it could be forever. Do you guys remember what cells don't divide anymore? Like you only have so many. Which cells are those? Can you guess which cells you only get so many? Say it again. Brain cells. So if you choose to do something silly like drugs or drink alcohol, you hear me? They kill your brain cells and you need them, okay? Or if you get into an accident and you kill brain cells, they don't come back, okay? So they never divide again. There's one cell that goes from the back of your head all the way down to your bottom. What is that one called? Ooh, there's just one cell from the back of your head all the way down to your bottom. What is that one? Nerves. It's one big nerve cell. What if you damage that nerve cell? Or what if you kill it? Do you get another one? No. And so you're going to be what? Paralyzed. So those are cells that hang out in the G0 phase. They never divide again. So at G1, the cell has to decide whether it's going to hang out in the G0 phase and not divide. But if it decides, you know what, I think I will divide, I think it's appropriate for me to do so, then it's going to get a little signal. It's going to say, you know, here's my signal to divide. At that point, it's got to copy its chromosomes during the S phase. Okay. After the chromosomes are copied, and we've already talked about how DNA gets replicated, right? We went all into detail. Helicase unzips and unwinds, and DNA polymerase adds the complementary nitrogen bases. We've done that. So that all happens during the S phase. Then after that's all done, we go to G2 or second gap where the cell grows some more. And at that point, we're ready to go on to mitosis. Okay, so all of that occurs during interphase. That's the reason why the cell spends 90% of its time there, because that's a lot. That's a lot going on. We talked a little bit about S phase, but we kind of already knew what's happening. DNA gets replicated, dividing cell replicates DNA so that it can put the exact same DNA in both of the new daughter cells. Okay, so separate the DNA correctly in both of the new daughter cells. We talked, we should be about right here, yes? Chromosomes? So if this is my DNA, and heads up, make sure you're paying attention, because students sometimes can't visualize how this works out. So here's the DNA, and we said that DNA was double-stranded, right? So two-stranded. Now, here's what happens. That double-stranded curly molecule gets wrapped around proteins, and then those proteins get squeezed together, and then they get squeezed together even more. At this point, this DNA plus protein is what we call chromatin. That chromatin squeezes together a little bit more, all the way until you get a big chromosome. Okay, so DNA is organized into chromosomes, wrapped around proteins. That DNA plus protein complex is called the chromatin. And then it gets condensed further. And at that point, we call it a chromosome. So essentially, a chromosome is just DNA tightly packed and wrapped around protein. Any questions about that? So I think we can all agree that there must be a ton of DNA in one chromosome, right? Since it's all so packed tightly. Also, question, how many chromosomes do you have in a regular body cell? Yeah, okay. It's just one number. Not 1,000, good Lord, that's a lot. It's 46. 46 chromosomes in a regular body cell. That having been said, I said body cell. I didn't say egg or sperm cell. So if you're a girl, you have eggs, and if you're a boy, you have sperm, yes? We kind of know this? Birds and the bees? Okay? When egg and sperm come together, you make a freeloader. Anyways, that being said, eggs have 23 chromosomes and sperm have 23, because 23 plus 23 equals what? Excellent, my pre-AP students. Good addition. All right. That's what they actually look like under the microscope. Kind of funky looking, huh? I know. All right, I don't think we got to this point. Or did we? Did we draw this? Okay, so if you didn't do so, if you did, bear with me. On the right side where it says diagram, I want you to draw this. Okay, draw that. Lord Jesus. All right, now I just told you you have 46 chromosomes, right? Yep. And half of those chromosomes you got from who? From mom, and the other half you got from? Awesome. So here's a chromosome you got from mom. So let's just pretend that's the mom chromosome. And then here's the dad chromosome. Let's say that these two chromosomes have the same genes on them. Okay, so these have the same genes. So here's the gene on mom, here's gene on dad, and let's say that this is the gene for widow's peak. How many of us have a widow's peak? I do. Do we know what a widow's peak is? It's like that kind of V hairline. Yeah. So let's say that mom gave you the gene for widow's peak and it's dominant, and dad didn't have it, so that's recessive. And so guess what? You have a widow's peak. So you can blame mom if you want to. But notice, even though these have different expressions for your hairline, they're still the same gene for hairline. You with me? You guys with me? So it's just expressed differently. So mom's gene is the widow's peak expression, and dad's gene is the not widow's peak expression. So if you get a chromosome from mom that has the same genes as the one from dad, we have a name for those chromosomes. Those chromosomes are called homologous. Write that down. Make sure you label it. What does homo mean? Same. Good. These are chromosomes that have the same genes on them. Now, they don't necessarily express the same way, right? But they have the same genes. One you get from mom, the other one you get from dad. Sweet. Now, after S phase, so go ahead and write S phase on top of this arrow. What happens after S phase? What is the purpose of S phase? To copy the chromosomes. So what happens is that the mommy chromosome doubles and the daddy chromosome doubles, right? So we're going to double up on our chromosomes. Okay, so we go from single-stranded, please write that. We go from single-stranded chromosomes to, guess what? What are these? Double-stranded. Because they just got copied. How are we doing so far? We're good? Now, in the middle right here, what keeps them together is called the centromere. So right there. It keeps the two copies together. So now, instead of being called chromosomes, each one is now going to be called a sister chromatid. Write that down. So each one is now called the sister chromatid. Okay? All right, so just so you kind of understood this. Now, go back up to your cell cycle. I want you to add some stuff to your cell cycle real quick, and it's going to become very important at the very end. Okay, so on your cell cycle at the gap 1 phase, I want you to add the G1 checkpoint. Okay, so at G1, there is a checkpoint. The checkpoint is where the cell stops and decides whether or not it is appropriate for it to divide. Okay, now let's say it's a cell that goes into G0 or stays in G0. Again, what is G0? No dividing, either temporary or permanent, like brain cells. Okay? If the cell is one that likes to stay in G0, if it's a cell that likes to stay in G0, then it's not going to keep going. It's going to decide, you know what? I'm going to stay and I'm not going to divide. But that's at checkpoint, right there, checkpoint. There is a second checkpoint at G2. That G2 checkpoint determines whether or not the DNA has been copied correctly during S phase, and if it is now prepared to go to mitosis. Now, in a minute, we're going to go over why or what happens if these checkpoints are ignored. There is a third checkpoint, and I want to go over it a little bit later. Not right here, right now. But for right now, I want you to add to your cell cycle the G1 checkpoint and the G2 checkpoint. Shayla, you good? Did you write this down? All right, so now we're going on to mitosis. Go ahead and flip. Okay, mitosis is just a division of the nucleus. You can write that down. Division of the nucleus. That's the easiest way to remember it. Division of the nucleus. So we're going to divide the nucleus into two. Now, we've already copied DNA, right? We have these double chromosomes. We've copied DNA. We're good. Now we need to just separate them
I put the same number of chromosomes in one nucleus and the same number in the other nucleus, and then separate the two cells into two daughter cells. Metaphase, anaphase, telophase. So we're going to go through each one. So just as your definition for mitosis, just write division of the nucleus. That's all it is. It's division of the nucleus. I'm not going to make you draw that because you're already going to draw it. Okay, so it seems unfair to make you draw it twice. So go straight to prophase. What does pro mean? It means first. So prophase is our first phase. And if it helps you remember mitosis, it's PMAT, prophase, metaphase, anaphase, telophase, PMAT. Okay, so first things first. I want you to draw the cell in prophase. I want you to draw the cell in prophase. And I want you to label it. Some things I need for you to notice. First of all, I want you to make sure that your nuclear membrane is broken up. Because during prophase, the nuclear membrane starts to break up. I also want you to put like an X mark through your nucleolus because the nucleolus disappears. I also want you to notice that the centrioles start to move and they start to kind of extend these spindle fibers out. Start to get the spindle fibers so you can see that. And you also notice that the double-stranded chromosomes are really condensed. So to me, they kind of look like worms inside the nucleus. So you can see that a whole lot better than you do in interphase because they're not as condensed in interphase. So on your test, it's really important that you know what happens during prophase, like each phase. So including interphase, you want S and G2, but make sure you also know what the cell looks like, what is there and what is not there. So again, in prophase, the nucleolus disappears, the nuclear membrane starts to break down, the chromatin is now very condensed. You start to see the spindle fibers and the centrioles.
You guys need another minute? Is that a yes? Okay. Say it again. Oh, there it is. Okay. So prophase is first. Starts with pro. So that means first. And then we go on to the next phase. What's the next phase called? Metaphase. What does meta mean? Anybody know? It means middle. It means middle. So I want you to look at this one. What's going on? Can you describe it? The chromosomes are lined up in the middle, right? Yep. So go ahead and draw that. Also notice there is no nuclear membrane. Right? There is no nucleolus. The centrioles are now at the poles, the north and the south. And the spindle fibers are attached to the centromeres of each of the chromosomes. Okay? So chromosomes line up in the middle. And then the spindle fibers coordinate the movement. That's metaphase. To me, that's like the easiest one to remember because they are in the middle.
Okay, what's next after metaphase? Anaphase. Okay, first draw it. What does it look like has happened? Separated. Very good. So your sister chromatids are being pulled in opposite directions. So being separated and pulled in opposite directions. Don't forget, this chromatid has the exact same information as this one. So that means that all of these are going to be in this new cell, and all of these will be, or these chromosomes will be in the other new cell. Okay? Now, we want to make sure that these sister chromatids are separated properly. In other words, what we don't want this chromatid here to get stuck to this one. So if it gets stuck to this one, that means that this cell will have an extra chromosome and this cell will have one less chromosome. Does that kind of make sense? Yes, no, maybe so? Let me know. So we want to make sure that the chromosomes get separated correctly. And because of that, this is our third checkpoint. Can you write that down? This is the third checkpoint. Write that down and also put it on your cell cycle diagram on the other page. So third checkpoint is in the middle of mitosis. That's where your third checkpoint is, where the cell is going to stop and check to make sure that everything has been done correctly. Okay, so the cell's going to check and make sure each chromatid was separated correctly and not one got stuck to another or a piece of one gets stuck to the other. That could be very, very bad. And we're going to talk about what happens when that happens. Like that could be very, very, very bad. So this is that third checkpoint where the cell stops to make sure that it's doing everything correctly. Okay, after you've added that to your cell cycle diagram, go back to anaphase. Sister chromatids separate, move to the opposite poles, and they are pulled at the centromere, so right here in the middle. That's anaphase. Okay. All right, so basically, I'm just showing you how the chromatids are separated. Here's the centromere. Okay, here's chromatid 1, here's chromatid 2. Remember, they're double-stranded because we've gone through S phase and they've replicated. Okay, so, and then they just get separated going into the opposite sides. This chromosome is going to go into one daughter cell, and this chromosome is going to go into the other daughter cell. And that's it. All right, telophase. Last phase of mitosis. Go ahead and draw it. I know that's going to take a minute, but go ahead and draw it. Notice that the nuclear membrane is starting to come back. And also, you notice that the nucleolus is starting to form again. And that's in each of the new cells. You'll also notice that there are no spindle fibers. Spindle fibers are gone, but you do still see the centrioles, but only in animal cells. Only animal cells have them. So make sure you draw that and label it. Again, the biggest trick to this is knowing what's happening during each phase and being able to identify a picture of it.
Okay? So chromosomes arrive at opposite poles. You're going to get two new nuclei. We call them daughter nuclei, daughter cells. The nucleoli come back. And you do not see spindle fibers ever again until the next time it starts to divide. Okay? Now we're going to start cytokinesis. So mitosis is done, prophase, metaphase, anaphase, telophase are done. Now it's time to do cytokinesis. Quick definition of cytokinesis, and it's not on the screen, so you need to write it down. So it is the division of the cytoplasm. So cytokinesis is just the division of the cytoplasm. Let me put an S there. Division of the cytoplasm. So it's division of the cytoplasm. That's all it is. All right. Animal cells and plant cells do it differently. So on the left side, you're going to write how animal cells do it. And on the right side, you're going to write how plant cells do it. Okay, so in animal cells, you get what's called a cleavage furrow. And I'll show you a good picture of that, because that one right here, do not draw that again, because that's a lot to draw. All right, so here's what this looks like. That cleavage furrow looks like that. And here's an actual picture of it under microscope. It looks pretty gross, actually. So that's what your cells look like when they divide. That's what animal cells look like. And that's the end of cellular division. So right when those two cells are separating, right? So that's cytokinesis, that division of the cytoplasm, when two cells are finally, or the one cell is finally becoming two. So that's in animal cells. Plant cells look different. In plant cells, you get what's called a cell plate. And so that's what it looks like. So a cell plate in plants and a cleavage furrow in animals, which is weird, I know.
All right. Flipping over from cytokinesis. So we're done. We've gone through interphase, G1S and G2. Gone through mitosis, prophase, metaphase, anaphase, telophase. We've gone through cytokinesis, the finality, the division of the cytoplasm. We're done. That's what it actually looks like under a microscope. That cell plate in a plant cell. All right, so this is what mitosis looks like. This looks like probably a shark cell. I feel like I've seen this one. Nope, plant cell. I lied to you. Sorry. So here's interphase. Everybody knows how the chromatin looks kind of bunched up there. Prophase, it gets condensed. We're going to skip prometaphase. And then in metaphase, it kind of looks like stitches. The chromosomes are all lined up in the middle. Then anaphase, the chromosomes are being pulled. And then telophase is the end. So, help me out. Let me see if you guys were listening. Where would you say this cell is? Your choices are interphase, prophase, metaphase, anaphase, or telophase. You say pro? You're close. It's before pro. What's before pro? Interphase. Now, one trick about interphase is that notice how you still have a nucleolus. Do you guys notice that? It's like a black dot in there. There's the nucleolus right there. And sometimes you can have multiple nucleoli. So that's one way that you can notice. And also, you still have a nuclear membrane. Okay. Also notice that the majority of these cells are in interphase. What did I tell you about interphase? The cell spends most of its time in what? Interphase. What percentage of time is spent in interphase? 90%. So when you're looking at cells, more times than not, you're looking at it while it's in, guess what? Interphase. So all of these here are going to be interphase, right? Does that kind of make sense? All of these are interphase. All of these are interphase. Now, let me give you a different one. I'll put it in a different color. Let's try this guy here. Definitely metaphase. Metaphase kind of looks like stitches, right? So it looks like the chromosomes are all lined up in the middle. Good. All right, let me try a different one. Let's try this guy here. That one's definitely anaphase. You can see the chromosomes being pulled apart. Let's try this guy here. Say it again. That one's kind of tricky, huh? Telophase. You can start to see the nucleolus come back. You can see the nuclear membrane come back, but it seems as though these cells are way too small, right? Like they're very small. So it looks like they've recently finished dividing. Does that kind of make sense? So on your test, you'll see this, maybe, and I might point to one and say, okay, what phase is this cell in? Got it? All right. All right, so evolution of mitosis. I don't want you to write much here. Prokaryotes or bacteria, they don't go. Why? Because they don't have a nucleus, right? So they go through what's called binary fission, which is the precursor to or what mitosis evolved from, right? Because we talked about
prokaryotes, and we said that prokaryotes were most likely the first organisms on this planet, and so that we all evolved from bacteria. That being said, binary fission is very simple. It is not as complicated as what we just went through. Binary fission, you've got one circular chromosome. That one circular chromosome copies itself. The cell gets bigger, divides. That's it. We're done. Question. Do you think it's faster than mitosis? Yes or no? What do you think? Yeah, much faster. Bacteria can actually double a number in 20 minutes. So that means that every 20 minutes, the bacteria in your mouth double. So by the end of the day, when it's time for you to go to bed, what are you supposed to do? Brush your teeth. Why? Because by the end of the day, you have a ton of bacteria on your teeth because they double every 20 minutes. Just putting that out there. All right, so that's binary fission. Let's talk about cancer. What is cancer? Somebody tell me. What do you know? So mutation. I love it. Good job. All right, so cancer is a cell division control. So remember when I was telling you to put all those checkpoints right there? So checkpoint here, checkpoint here, checkpoint here, where the cell stops and says, you know what? Is it okay for me to continue? This might not be a good idea. I need to check and make sure everything's all right, make sure everything's working, make sure that I've copied DNA perfectly, make sure that each new cell is going to get the same number of chromosomes. What if these checkpoints don't happen? What if the cell ignores its checkpoints? Then that means that a cell could potentially divide uncontrollably over and over and over again. Write this down. Uncontrolled cell growth, uncontrolled cell division. Cells just divide and they stick to each other. You know, typically cells don't necessarily like to stick to one another. There's only a couple of cells that like to be next to each other. But usually cells like to have a little space from other cells. But these cells, cancer cells, are like, hey, it's all a party. Let's all stick to each other. And I think most of us have had some experience with either a family member or someone we know real close that has suffered with cancer, right? We just didn't really know what it was. Okay? I'm not writing all that down. But write this down. So unlimited growth, checkpoints are ignored, when I say escape apoptosis, we talked about apoptosis before. That's a kind of a planned cell death. Cells kind of already know when it's time to die, and so they might tap their lysosome and say, hey, lysosome, it's time for you to explode and kill me. So it's kind of like a planned cell suicide. But in cancer cells, that apoptosis, that plan is not enacted, like it just doesn't happen. So you get this immortality where cells divide constantly and they never stop. I want to say, I forget this lady's name. I think it was Henrietta, but I don't remember the last name. But this lady, she's a black lady, like the 40s or 50s, and she had a particular form of cancer. And what the researchers did is they took her cancer cells and they used her cancer cells for research. And to this very day, her cancer cells still divide. Like, it's almost like they're immortal. It's crazy. And of course, she's been long dead. At some point, your body says, you know what? I see this clump of cells over here. It looks hungry. When your body notices that, sometimes your body will send blood vessels to feed the cancer cells, with oxygen and glucose. So sometimes your body will do that. What do you think happens when a clump of cancer cells starts getting fed? What do you think happens? It gets bigger, and then some of those cancer cells may break off, go into those blood vessels, and then now they're someplace else in your body dividing like crazy too. Okay, and so that's when we say the cancer has spread. So I want you to write down what can cause mutations that ultimately cause cancer. Radiation, really any form of radiation. You guys remember we talked about Rosalind Franklin? The lady who took an X-ray picture of DNA. Remember her? What happened with Rosalind? Yeah, she died. She didn't know that X-rays cause cancer, and so she died of cancer. So lots of radiation exposure. We watch like Marvel movies and we see the Hulk and we're like, oh, gamma radiation makes you into the Hulk. Nope, gives you cancer, and then you die. It's not pretty. We know that cigarette smoke, and guys, not just cigarette smoke, okay? Marijuana smoke. I know people don't really recognize it, but it's true. Any type of smoke that you're inhaling, as well as vaping, even though it's water vapor, it doesn't matter. It doesn't matter. All of these can trigger mutations, and those mutations can cause all the things that lead to cancer. So here's a tumor, right? So we just said that the cancer is kind of basically just cells that have ignored their checkpoints or decided not to die or what have you. So now they're all clumped up next to each other. Then the tumor starts to grow and it starts to invade other tissue. And it could also start to kind of sit on nerves. And that's where that pain comes from. And so now this tumor is kind of growing. Notice how over here the tumor is nice and round. This is the time when you could probably get it removed, right? So this is the time when you could probably have surgery. Here it gets a little bit more complicated, right? So surgery is a little bit more difficult. But when I get to this one, what has happened? What are you noticing? What are these? Those are blood vessels, right? So your body has recognized this tumor. It's like, oh, these are a group of cells that perhaps need some food. And now your body is starting to send blood vessels with food and oxygen. And so what happens to the tumor? What does the tumor do? It starts to get bigger. In addition to getting bigger, now some of those tumor cells break off, go into the blood vessels, and now they go everywhere else. And that's when we say the cancer has what? Spread. Does that kind of make sense? Sort of, kind of, maybe? Okay, go ahead and write down benign tumor. A benign tumor is just a group of abnormal cells, and it stays in that spot. So right here is that benign tumor. It just stays in that one spot. My dog has one. He's got like a little tumor. It's really on his butt. It's weird, but whatever. But, you know, it's just kind of sitting there. And I was like, okay, no big deal. We're gonna get it removed. No biggie. When it gets bad is when that tumor leaves the original spot, right? And the way that it can do that is if there are blood vessels that feed it and it's able to travel through those blood vessels to other places. Okay, that's going to impair organs in other parts of the body. And so if you ever wondered why it is that people die of breast cancer with Miss Johnson, how can you die of breast cancer? It's because those cancer cells will leave the breast tissue and go someplace else. Does that kind of make sense? And so it can go up to the brain, the liver, or other places and begin to grow in other places. Okay, so that's a malignant tumor. All right, give me some treatments for cancer. What are some things we can do? Chemo, good. What else? We've got radiation and chemotherapy. Another treatment is surgery, right? Surgery. So write those down. Surgery, radiation, chemo. I read that there is a new kind of treatment for kidney cancer where the researchers have found out that they can reprogram a person's immune system cells to target cancer cells. So using your own immune system to kill the cancer cells is kind of cool. So treatment, radiation, chemotherapy, surgery. Take a picture. Or, I'm sorry, turn in your notes. Okay. So, you have a choice. You can do your foldable in class, yeah, or you can do your foldable at home. For your foldable, you can watch the Media Sisters videos of the cell cycle mitosis. You can watch the Media Sister videos. Or you can just use your notes. It's entirely up to you. The Media Sister videos are kind of cute. So sometimes that's helpful.
foldable template. And this is a Johnson foldable. It's gonna have something. Just fold it just like this, where you see the circle in front. So essentially, this one looks just like a review of everything you just did. So you're going to draw and label the cell cycle in the front. And then you're going to draw and describe each phase. Okay, draw a label. So don't forget to label. Okay? Again, you can use the notes that we just did, or you can use the Meet the Sister videos that I put on the camp. So you can use either one, and the Meet the Sister videos are easier to understand. That is a good option. Next time, we will put Play-Doh, and we'll go through each of these phases with Play-Doh. And hopefully by that time, I'll be like, well, this is easy. Again, your test over this information is next Friday. So you guys have 24 minutes, if you want to, instead of doing this for homework. You can do this right now. Okay? So you can have to use your notes, or you can watch the video. All right,